To introduce myself, I am Clarissa Czekster. I'm originally from Brazil, but I've been in the UK now for 7, almost 8 years. The work that I do is related to finding new drugs to treat disease. We're specifically interested in microbes and antimicrobial development. We also do some work looking for molecules in nature. And because we're looking in nature, we don't know exactly what we're going to find. And we have some work related to anti-cancer compounds, for example. But the majority of our focus is on microbes. This work has been a great collaboration with Megan Burke-Castle in Dundee and Marcus Bishop here in St. Andrews. And my group, what we started doing was we wanted to understand how this protein works. Because when Pseudomonas aeruginosa grows, it forms these communities called biofilms. And biofilms are very difficult to eradicate because they are thick and complex, and also bacteria are not particularly highly metabolically active when they're in a biofilm. So traditional antibiotic treatments are not very effective. We know that this protein, PAP, the aminopeptidase, is produced at high levels when bacteria are in biofilms. So what we wanted to know is how can we affect the activity of this protein and what impact would that have in biofilms? So we started with a very basic science approach. We just wanted to understand how this protein works, because when bacteria make the protein, they make it in an inactive state. So the protein is almost not doing anything, and it needs to be activated extracellularly for it to become active. And this is part of the regulatory pathway that naturally occurs when this protein is made, because bacteria don't want to have an active peptidase that can just degrade everything unless they really require this activity. So we started out understanding basic principles about how this protein works. And our work revealed that this protein has a specific mode of activation. And we explored and exploited this mode of self-inhibition activation to produce compounds that we could add and recap this self-inhibition mechanism. And this has consequences for bacterial survival in a biofilm. And this is something that is very important for us, but also for society, because we don't currently have drugs that target biofilms specifically. Yeah, so Pseudomonas aeruginosa is one of the organisms that the World Health Organization has flagged up as one of the priority organisms that we have to develop new drugs for. We are interested in Pseudomonas aeruginosa because it's a big problem in chronic infections. So there is a need to develop new treatments that are specific for this kind of infections. So, for example, patients that have cystic fibrosis, but also other pulmonary chronic infections, they spend their whole lives undergoing treatment for bacterial infections. And if you spend enough time being treated for disease, you do reach a point where not only you're suffering for all the side effects, but you run out of drugs to use. So we have to develop new drugs to fight these chronic infections. So there is this motivation that is, a real life problem that we have, that we must develop new antimicrobials that target these kinds of infections. But also from a basic science point of view, Pseudomonas aeruginosa is a very interesting organism, which is kind of a problem to us humans, but it's very interesting because it can survive in so many different environments, and it's very adaptable. Because it's very adaptable, it has many different strategies to cope with a lack of almost any nutrient that you can imagine. So it's very resilient to fluctuations in the environment. And this is one aspect that we want to understand better and hopefully control this and, stop it from being so adaptable so we can fight these chronic infections. We need new treatments of all kinds. That includes small molecules, larger molecules, everything. But also, when you think of some antibiotics that are currently used, like vancomycin, for example, it's a very big peptide molecule. So some antibiotics that we already have that are derived from natural products or inspired by natural products, they are peptide-based or have a peptide component to them. So it's not necessarily a completely, out there idea. There are some advantages. A lot of the work that we do in the lab is focused on peptides because peptides can mimic some interactions that naturally occur. So that's an opportunity to design compounds of your interest. And also because over the past maybe 10 years, we became much better at making peptides, both chemically, but also using cells and using enzymes, which is some of the work that we do in the lab as well. We have a lot of different ways to make peptides of our choice. And because of that, we can tailor these molecules to be what we want them to be. And we have more control over how they're being made, if that makes sense. We don't necessarily always rely on finding a natural product and on nature to make these compounds. But the fact that they can mimic a lot of interactions that naturally occur is desirable because it's a property that we can explore, which is what we did in this work. But it is a relatively common field of interest in peptide, disrupting protein-protein interactions, for example. Peptides are very attractive. It's absolutely vital for this work because the work that we do in my lab is very centered on enzymes and biochemistry of proteins and understanding how they work. So we look at their structure, we make modifications on the proteins themselves, and we understand them. But if we want to put this into context, we need to look for collaborations for, to be able to do a lot of experiments and answer many questions that we couldn't really answer alone all by ourselves. So the collaborations that we have in this particular project are very diverse, and it was really great for Chris, who works on this project, and to me to be involved and soak some of the knowledge that came from Megan and Marcus. And they are individually also very different in their areas of expertise. And I think it's pretty cool that we are all brought together by a common, interesting question that bring together the expertise that they have. So Megan is a molecular microbiologist. So she does a lot of work with genetics of microbes and looking at the effects that an antibiotic can have, for example, in resistance, but also generating variations of a particular microbe that don't have a certain protein of interest. And this is important for us because we can explore what is the effect that completely removing this protein, for example, from Pseudomonas aeruginosa have on biofilms or on growth. So we can bring together the results that we get with the biochemistry and the peptide inhibitors with mutants and variants of Pseudomonas itself. And that's very powerful to validate how we think the peptides are working, but also to explore what kinds of effects we can see. And Marcus's collaboration was also vital because Marcus knows everything there is to know about microscopes. So he was fundamental in imaging biofilms and analyzing the data properly, but also figuring out what we can and what we cannot do with the kinds of experiments that we wanted to do. So yes, I'm really happy that we got together to do this work. And it was the first time that this group of people collaborated together. And I hope that we'll keep producing interesting research in the future. It's actually almost too exciting because there's too much to do. I need to try to, focus a little bit to pursue one question at a time because this work opened a lot of questions. So we're currently working on follow-up. There's 2 follow-up projects, but also new grant applications as always. And some of the key areas that we want to focus on some are more applied, and together with industrial partners, we want to see how this peptide can be delivered. We are interested in the aspect that it's an extracellular acting molecule, so it doesn't need to penetrate cells. And that opens a lot of possibilities in how you could deliver this particular peptide or variations of this particular peptide. So this is like a practical aspect that we're looking into. We're also pursuing questions that are related to basic biology. How is this molecule actually killing cells in a biofilm? We don't completely understand that, and we want to understand that better. We also are interested in, are pursuing ways to test if this is a common strategy that we can use for other proteins, and how feasible is that? And, is that a conserved mechanism of self-inhibition, for example, that we can explore?